Hello, students. Welcome back to our next topic. And this week we are continuing with this multi-week theme of cells because there is just so much to talk about when it comes to cells. So last week we talked about cell processes, some of the basic functions that take place within the cell. The week before that, we talked about the cell structure and some of the organelles. Well, we are continuing with this theme of cells this week once again as we dive into the topic of the week, cell growth and division. Cell growth and division. So take a moment, if you haven't done so already, to get your books open and turn to your lesson notes page on the topic cell growth and division. While you're doing that, I have a question for you, and the question simply is this: are you bigger today than the day you were born? It's not a rhetorical question. I want you to think about it and answer honestly. Are you bigger today than the day you were born? I certainly hope your answer is yes, or I feel very, very bad for your mother. Of course, you are bigger today than the day you were born. But my follow-up question to this is why? Why are you bigger today than the day you were born? I know you're older, but really think about this for a moment. Why is it that you are bigger today than the day you were born? Is it because your cells have grown in size, just like you are growing in size? Is it because you have more cells in your body now than when you were born? Is it for some other reason? Pause me now and take a moment to discuss. Okay, I'm back. And here we are. We're going to continue this discussion of why you are bigger today than the day you were born. And hopefully you had some fruitful discussion there. But here's the answer. Cells grow in number, not in size. In fact, later this week, you're going to be doing a little lab activity where you're going to see why cells can't grow in size too much. There are limiting factors as to how big a cell can grow. But cells grow in number, not in size. The answer to the question, why are you bigger today than the day you were born? It's because you have more cells in your body than you did when you were born. In fact, if you go back far enough, you all started out from just two cells, a mama cell and a daddy cell. And when your mother and your father loved each other very much, those two cells came together to create a fertilized egg, which really was one cell. And from that one fertilized egg cell, it started dividing over and over and over again until eventually you got to where you are now, where your body has trillions upon trillions of cells. I couldn't tell you exactly how many cells are in your body because I'm not about to count them all. I don't think anybody really could. But you've got trillions of cells in your body right now. Now, when you get older, you start losing more cells than you make, and maybe some of you have noticed that older folks tend to get shorter. They actually lose height. Part of that is the cellular regeneration rate is being reduced, and they're starting to lose more cells than they had when they were your age. But anyway, cells grow in number, not in size. That is a very, very important distinction to make. But let's pick upon this idea a little bit more. How do they do this? How do they grow in number? How is it possible that there are so many more cells in your body today than the day you were born? Again, we're starting with one fertilized cell into trillions of cells by the time you're your age. How in the world is that possible? Well, it is possible through our one topic here of this week, cellular division. Cells do this by dividing, and cells have perfected this division process very well. We're going to be spending a lot of time today on the steps of cellular division, but let's just say that cells are experts at dividing themselves over and over and over again. So, as I alluded to earlier, cellular division is something cells do over and over again. And it doesn't just happen like that. It actually takes place over several phases or several steps. So there are several steps or phases that we are going to be discussing today in the context of cellular division. And if you look at the background picture here, you'll see the steps summarized here from one cell to two cells, and we're going to talk at length as to how cells go from one to two cells. It's going to take us a lot longer to talk about these phases of cell division than it actually takes cells to perform it. And we are going to be watching it take place on Thursday in the lab as well. So let's get right to it, and let's begin with a basic diagram of some of the key parts to a cell. You know from a couple weeks ago that there are a lot more parts to a cell than this diagram shows, and that is absolutely true. But we're going to focus today on the nucleus and the chromosomes because these parts of a cell are crucial when it comes to understanding how cells divide. So what I'd like you to do in your book is this. I have provided space for you in your book to draw some different diagrams. This diagram can go on the very top of the page as a general labeling diagram to illustrate the cell itself by a big circle and a smaller circle inside called the nucleus. And you'll notice these V-shaped structures, which I'm calling chromosomes. Now, even though we're going to be talking about cellular division in the context of human beings, really, cellular division takes place with any living organism. And there are different numbers of chromosomes for different species of organisms. For instance, human beings, we have 23 pairs for a total of 46 chromosomes in the nucleus, every nucleus of our cells of our body. We're not going to draw all of those chromosomes because we'll lose sight of the forest through the trees, or lose sight of the trees through the forest. I'm not sure how that expression goes. By the way, is that an idiom? I'm not sure what kind of an expression that is. But so here's the deal. We're going to have in our diagrams 4 chromosomes. We're just going to limit it to 4 chromosomes. So what species could this be? Maybe a fruit fly? I don't know. But we're going to limit our diagram to 4 chromosomes, and these V-shaped structures will illustrate those chromosomes. We don't need to get bogged down in the exact number because it's the process, it is the phases, it is the steps that we are most interested in. So with that basic diagram understood, let's begin with phase number 1. Phase number 1 is called interphase. Interphase is the first phase of cellular division. And the prefix inter basically means between two things. Have any of you ever been to like a theatrical production of some kind, gone to the theater? And I'm not talking about the movies, I'm talking about a live production of any kind. Maybe you could share with us some of those productions that you've been to. Well, I've been to a number of productions, and some of them can be pretty long. And so, between the first act and the second act, if they're quite lengthy, they might have what's called an intermission. And the intermission is a time period between two major time frames of a play, let's say. And it gives you a little break to uh, excrete your wastes, allow your cells to undergo some metabolism, respiration, etc. It gives you a little bit of a break before you have to sit down again. But interphase is basically a phase that takes place between other phases. So let's expand on this a little bit. During interphase, something very important takes place. It's not that nothing is happening. Something very important is happening. It's just not dividing yet actively. And what is happening, illustrated by this diagram, is that the chromosomes are copied. The chromosomes are being copied. Now, after you've written that down, the chromosomes are copied, I want you to draw this illustration in your book as well, paying particular attention to what's happening inside the nucleus. If you recall from our previous diagram, the chromosomes were illustrated by little V's. Well, if you hold up a V to a mirror, it gives you a mirror image of that V. And so you have two V's, one V this way, one V this way. A mirror image of both of them makes, and I know it's tough to see through my hands, but maybe you'll get the idea that two V's point to point form an X. And what you're drawing here are really not four X's, but they are four copies of the original V's. So while you're drawing the chromosomes here, think about that. You're really drawing four V's with four copies of V's facing the opposite direction. So they look like X's, but they're really a total of 8 V's. They have been copied, but not divided yet. How many of you have ever made a photocopy of something? You receive papers all the time, notices that go home perhaps that have been photocopied in the office to send home to you or your families. A photocopy, a copying machine, does what we are talking about here in Interphase. Imagine putting a piece of paper on the copying machine. You push the copy button and boop, 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 out churns the exact copy of the original. It is a photocopy. That is basically what's happening during interphase. You're creating an exact copy of the original chromosome. So that's what takes place during interphase. Now, here's a picture, and I have to apologize that there's a hyperlink in the way here, and I should have removed this hyperlink. Oops. But that's not going to do it. So I'm not going to take the time to do it right now, but later on I'll show you this picture again. But you will be able to see under this hyperlink, and if you just view this PowerPoint online, you won't see this hyperlink shown here. But you'll see a picture of a nucleus undergoing interphase. By the way, this one up here is also going through interphase, and you can make it out by the fact that there's there appears to be a lot more activity or density within the nucleus. Here is just a normal nucleus, nothing is going on. Over here, you see a greater concentration of material. That's actually the chromosomes being copied within the nucleus itself. So that is a picture of interphase committed to memory. All right, let's head on now to prophase. Prophase, and I'm trying to use alliteration here too. Prophase is pairing up. So remember, prophase, pairing up. Think of PP. Think of PP when you're thinking of prophase. Prophase is the pairing up of the chromosomes, and it's the phase during which mitosis actually begins. Speaking of mitosis, pardon me for a moment. But mitosis. Mitosis, not mitosich. But mitosis is a vocabulary term, and hopefully you've defined it and are familiar with the meaning of mitosis. Mitosis is specifically nuclear division. Now, that doesn't mean like a nuclear power plant is going to explode here. We're talking about the nucleus inside a cell. So the nucleus begins to divide during mitosis, which is one of the things that happens during prophase. And there are a series of things that take place during prophase that we can observe. So let's get to it. First, a spindle forms. What in the world is a spindle? Raise your hand if any of you know what a spindle is or whether you've heard that word used before. Maybe you were thinking of a Disney movie, Sleeping Beauty. Sleeping Beauty, there was a spindle in that movie. Perhaps you saw that movie. And that movie involved a curse on Aurora or Sleeping Beauty. Didn't she have another name in that movie as well? I can't remember. Sleeping Beauty, Aurora, Princess Aurora. I think she had another name too, didn't she? Anyway, if you look at this spinning wheel, there's a part of the spinning wheel, and I don't want to touch it myself. Hopefully I won't fall asleep. But there's a part of this spinning wheel called the spindle. This is the spindle of a spinning wheel, and it's got a really sharp point to the end of it, which is what Aurora should not have touched on her 16th birthday, or she cursed the whole place, the whole kingdom, right? Well, so how does this relate? I know we're talking about Disney movies. What's up with that when we should be talking about cell division? Well, there is a connection, because this structure here called the spindle can also be taken off, and actually people who spin, yarn, and thread, they're familiar with these terms because this is a handheld spindle. And this handheld spindle, you get a clump of wool from like a sheep or an alpaca or a llama or something that has fibrous fur, and you twist that fur, attach it to a spindle, whether it's on a spinning wheel or a hand-spun spindle, and you spin it. And when you spin the spindle, what it does is it draws these fibers into the thread or into the yarn. A spindle will take fibers and gather them together and start pulling them apart. This is the background, the history as to why biologists call the structure in the cell the spindle. Because when the spindle forms, it is literally pulling things apart. So remember, the spindle pulls things apart. And I'll show this to you in a diagram in a moment. But because a spindle forms, in fact, two spindles form and starts drawing things apart in opposite directions, the nucleus itself, which is surrounded by the nuclear membrane, remember that filter paper from a couple weeks ago? Well, that nuclear membrane starts stretching bigger and bigger and bigger until the membrane starts to tear, rip, and break apart. I don't know if any of you know what a stretch Armstrong is. Do any of you know what a stretch Armstrong is? Well, when I was younger, they had just come out, like back in the early 70s, I think the Stretch Armstrong came out, and my older brother had one. And even as a young kid, I was always a curious scientist. And I wanted to find out, because the commercials all said you could stretch Armstrong indefinitely, basically. It sounded like from the commercials, you could just keep on stretch Armstrong, and he would never break. He would just come back together again. So as a little kid, I tried an experiment. I took a stretch Armstrong, and well, maybe I shouldn't tell you exactly what I did, but let's just say that I was able to stretch the stretch Armstrong beyond its limit, and his skin started tearing apart, and his insides oozed out all over. It's a mess that you don't want your mom to find, believe me, because it made a real sticky mess all over the place, as I remember. And I had to clean it up myself. It was not a pretty sight to see Stretch Armstrong stretched beyond his limits. But that's kind of what happens to the nuclear membrane. It's being stretched apart by the spindles forming and pulling apart, that the nuclear membrane starts breaking down. It's stretched beyond its limit. But more than that, something inside the nucleus is happening as well. The chromosomes pair up, and here's where that P comes in handy. Remember, prophase pairing up. Here's a key part of what's happening during prophase. Now, what exactly does it mean that the chromosomes are being paired? Well, have any of you ever gone to a dance? I know in the last year or so there haven't been any dances taking place because it's difficult to dance with one another if you are all socially distanced apart from one another. But imagine dancing. And here is a picture, a little animation of some square dancing. Now, I know years ago when I was younger, we used to actually have to square dance in one of the grades during phys ed. I think it may have been fourth or fifth grade. I don't remember exactly. But have any of you ever square danced? If you've never square danced, oh, it's a wonderful thing. And all you have to do is follow the instructions, which means you have to listen to the instructions in order to be able to hear the instructions in order to follow the instructions. But oftentimes with square dancing, you have to pair up with your partner. Then do-si-do, and around you go, and a wing swing your partner to and fro, yippity-ha, hee-ha. Anyway, square dancing, good analogy of pairing up with a dance partner. I know a number of years ago, we used to have a lot of school dances. Seventh and eighth graders used to be able to invite people from other schools, and you'd have school dances. And sometimes I'd chaperone these dances and stand there. And every year it was the same thing. You see all the girls on one side, all the boys on the other side. And very rarely would the boy and the girl pair up to dance because they were just too shy. But usually the girls would gather together in one big group and they'd all be dancing together and having a good time, dancing in the center of the floor. And all the boys would be standing around the outside, drinking some punch, talking to one another like, should I ask her? Shouldn't I ask her? I wonder if she's going to ask me. Maybe some of you know what I'm talking about. But pairing up. So we're not talking about actual dancing here, but we are talking about the chromosomes being paired up, getting ready to dance. Now, here is our diagram for prophase. And another way it helps me to remember which one is prophase when I'm drawing the diagram, and make sure you are copying this diagram down in your books as it's illustrated and labeled here. But another way that I remember which one is prophase is I think of a pig. Yes, I said a pig. I mean, use your imagination. Does this or does this not look a little bit like a pig? The way I have drawn it, especially since I've added the ears on the top. But, some of you, if you go to a dance, girls in particular, maybe boys, I don't know. You might put mascara and eyeliner and makeup on to make yourself look pretty. Well, here I envision a pig putting, is it mascara that you put on your eyelashes? I've never used mascara, so I'm not sure. And the eyelashes are very long because it's ready to go to the dance. And those eyelashes on this diagram are basically the fibers that are being drawn away from the nuclear membrane that is breaking down because of the spindles. Remember the spindles drawing apart the fibers of the nuclear membrane, which is causing the nuclear membrane to break down. And inside the nucleus, we have the chromosomes being paired up. They are lining up basically 2 by 2, getting ready for like a square dance because they are going to be dancing soon. Now, I know the diagram looks like a pig basically, but if we advance to our next slide, this is an actual slide of some cells undergoing prophase. And if you look at these two pictures here, this is early prophase and this is late prophase. And if you look at the little structures inside the nucleus, those are the chromosomes. And they are, in fact, pairing up with one another. And if you were to watch this take place live, and you'd see the images of the nucleus actually undergoing cellular division, you'd see how these chromosomes are moving into position to line themselves up, pairing themselves up, getting ready for a big dance party. So, we've talked about interphase and we've talked about prophase. Let's talk about one more phase here for today, and that is metaphase. Metaphase, think of the letter M for metaphase and think of the word middle. They both begin with the letter M. Again, a little bit of alliteration can go a long way in helping you memorize that metaphase is the middle of the cellular division. Now, what exactly does the word middle mean in this context? Well, let's find out. During metaphase, the chromosomes begin their actual separation. So here we have the cell, and the ends of the spindles are actually called centrioles. You might want to write that down in your notes while you're drawing this diagram as well. You can see the fibers on both sides pulling the chromosomes away from one another with the two centrioles. And what we've got here in the middle are all of the chromosomes have met in the middle. Baby, why don't we meet in the middle? Or something like that. Isn't there a song that says something about meeting in the middle? Meet in the middle. Meeting in the middle. The chromosomes are meeting in the middle during metaphase. Metaphase, meet in the middle. The chromosomes are meeting in the middle. And this has a lot to do with geometry. It has to do with aligning one another. All of these chromosomes are aligning each other so that there's enough room inside the tiny little cells, inside the tiny little nucleus, in order for them to undergo metaphase at this point. They've got to spread out. It's kind of like when I was younger, I used to play this game, Red Rover. How many of you have ever played Red Rover? Red Rover, send Bobby Sue over. And they'd have to run to the opposite side where your team was lined up in a line, and they'd have to run right through the line. It was one of those big games when I was a little kid. But you need a lot of people for that. But, we had people lining up. Well, here we have lining up chromosomes, and they're meeting in the middle. And here in this diagram, check it out. You can see that there is a clustering of darker red material. Those are the chromosomes, and they're starting to really line up in the middle. And you can see on either end are the spindle apparatus, at the end of which is the centriole. So here is metaphase. Metaphase, the next phase of cellular division. Do we have time for one more, maybe? Oh, golly, I don't know. Should we? Maybe we should call it quits there for today. I'll tell you what. Let's call it quits for the day at metaphase, and we'll pick it up again tomorrow with the remaining phases of cell division. So for today, I will say bye-bye.